So, yeah, I just want to give everyone kind of a little history and background so that we're all starting on the same page here. In the 1930s, PFAS were really developed for use as a non-stick water repellent coating. And it really, in the 1960s, aqueous film forming foam, or AFF foam, was developed. And it was developed after we had more than 130 sailors who were killed in the explosion on the USS Forrestal, and it burned for an extensive period of time. There needed to be a way to manage that fuel-based burn. Today, we see over 5,000 compounds in this class, and the uses include water and repellents in clothing and textiles, food packaging, personal care products, and of course, firefighting foams. So some of the possible health effects, these are just a few. It's not meant to be an exhaustive list. But you can see increased in cholesterol levels, changes in liver enzymes, small decreases in infant birth weights, increased risk of high blood pressure in pregnant women. We see decreased vaccine response in children, increased risk of kidney or testicular cancer, and several other like immunological effects in laboratory animals. And IARC Class 2B, it's been classified as Class 2B as a possible human carcinogen. There's just some lists of the health effects that are possible that we've identified. There is a number of recent legislative activities in this space. In fact, a total of 84 bills, amendments, and resolutions have been introduced into Congress since 2018. These pieces of legislation, they cover water supply, contamination, veterans and military exposures, health effects, and other pertinent issues. 3 pieces of legislation that are recent have related to firefighters and emergency responders. So I want to draw your attention to what's on the slide right now as some of these recent legislative pieces. You can see in 2020, a Senate bill was introduced and a House resolution was also introduced into Congress. And this particular bill was directing NIST to work with NIOSH specifically to study PFAS in firefighting PPE. And more recently, in 2021, we've seen a Senate bill introduced that's directing FEMA to work with the U.S. Fire Administration, EPA, and NIOSH to develop and issue guidance to firefighters and other emergency response personnel, goal being to reduce exposure to PFAS from firefighting foam and personal protective equipment, and to limit or prevent the release of PFAS from firefighting foam into the environment. Also, the purpose of this bill is to reduce the exposure to PFAS from firefighting foam and personal protective equipment. That's repeated. So those are the main legislative pieces that are out right now for consideration and really should be in the background of our mind as we're thinking through some of the activities we see presented today. Another thing that's important to recognize is these chemicals have been kind of colloquially termed as forever chemicals. They're water soluble, they're resistant to grease and oils, they have low volatility, they do not biodegrade, and they're often found in water supplies throughout the country at this time. Just a little bit of information about the actual chemical structure. I just want to draw your attention here to the top left. This first structure on the top left is octanoic acid, and it's a carboxylic acid with an 8-carbon chain. Now, perfluorooctanoic acid is formed when the hydrogens bind to the carbon chain. They're replaced with fluorine atoms. So this molecule is then known as PFOA or C8. The carbon-fluorine bonds are among the strongest bonds in chemistry. And these are the same, I just want to kind of point you here to the one on the right here, okay, to take a look at this. What you see here in this octanoic acid structure is that the carbon backbone is actually visible and the hydrogen atoms can be abstracted easily, allowing access to the carbon atoms. However, in PFOA, if you look at that structure here on the bottom left and bottom right, you can really see that the electron cloud of the fluorine molecules shields the carbon backbone, preventing side reactions, right? So we're talking about this limited backbone now that you have access to. And these molecules are stable and do not degrade naturally. And that's really why they're called forever chemicals. It's this right here, which you're looking at, what's happening in the chemistry that's protecting this backbone. So that's the overview we wanted to start with today, to orient folks to the health effects, how it's being used, what's going on legislatively, and kind of what's happening in the structure of the chemical, so that you understand why it's called a forever chemical. With that, we just want to thank everybody for coming today. We want to remind folks that quality partnerships enhance worker safety and health. Part of the mission for the Public Safety Sector Council through NORA is to make sure that we facilitate partnership and engagement through the dissemination of information and collaboration opportunities. So we thank all of our presenters for coming today to help facilitate that goal. We thank all of our audience members for being here today to also help us achieve that goal. With that, I'd like to turn it over to the first speaker.
So I'm going to talk today about some work we've been doing in New Jersey on looking at prevalence and predictors of PFAS serum levels among volunteer firefighters, and our thinking about how to sort of be getting at source apportionment to the exposure. And as Susan talked about in the introduction, there have been quite a few studies of AFFF exposure and PFAS exposure in firefighters, with evidence of increased levels of specific PFAS in firefighters that use these foams, and with years of firefighting exposure. Appropriately, most of this work has been done among career firefighters, who tend to be the firefighters who use AFFF. But as many of you know, over 2/3 of our fire service members in the US are volunteers, and they may be exposed to PFAS through AFFF, but there are other sources, as is mentioned, of PFAS exposure for firefighters in the PPE potentially, and then also potentially as they may be liberated from combustion during structural fires from those carpets and other household PFAS containing products. And of course, firefighters live in communities where there may be PFAS exposure through the water and dust. And so figuring out what the exposure may be is very tricky. And so our goal here, again, is to work with volunteer firefighters and assess prevalence and predictors of PFAS serum levels and sources of exposure. And we do this in the context of our research partnership with the National Firefighter Cancer Cohort Study that was begun by Drs. Burgess, Caban-Martinez, and Corkins, and continue to be leadership of this cohort and amazing research partners. So my group, the Cancer Assessment and Prevention Study, is a research partner of the FFCCS, and as such, we have been able to leverage the existing protocols. We do biomonitoring within volunteer fire departments who we partner with. We use the New Jersey Department of Environmental Testing Laboratory for PFAS analysis. They are a CDC biomonitoring program partner. And then we have adapted the National Firefighter Cancer Cohort Study survey for use in volunteers. And that survey collects information on demographics, occupational history, behavioral risk factors, as well as surrogates for firefighting exposure, such as years of firefighting. So what I'm going to do is give you some of the results from our 2 years of pilot work. And in that work, we worked with one fire department, I'll call Fire Department A. It's a large suburban volunteer fire department. We enrolled 135 firefighters, members of the fire department. Average age was about 40, almost all male, non-Hispanic, white, and with about 20 years of firefighting on average. And then I'll also, towards the end, talk about some of our work with additional partners through a FEMA funding, where we've enrolled more volunteer fire departments and have been able to do PFAS testing on members of 2 additional departments. So let me give you some results of our work with that first Department A. And again, we enrolled 135 volunteer members of that firefighter, and this work compares the non-Hispanic white males to non-Hispanic white adult males in NHANES in terms of PFAS prevalence or detects. And you can see for the PFAS that are usually measured in NHANES, the distribution is very similar, except for PFDOA, which in the latest NHANES cycle, there were no detects, and in our firefighters, we saw over 80% had detectable levels. We then looked to see whether there were differences in the level of PFAS in the serum, and we saw that 3 had elevated levels among our firefighters group compared to NHANES. Those were PFNA and PFDEA, which have been seen in previous studies of firefighters, and again, PFDOA, which has not. So we started to think, well, what's going on here? Are the source of these PFAS occupational or environmental, so from firefighting or other sources. And so we've tried to think about this a few different ways, and we're very early in the process, but the first thing we did is use the survey data to try to tease out some of those relationships. We looked at age, cumulative years of firefighting, numbers of firefighting calls responded to per year, and those are self-reported, and we looked at that in multiple linear regression with each of the different PFAS that were elevated. We saw that age was not associated with any of the PFAS levels, but PFDEA was associated positively with cumulative years of firefighting, and PFDEA with calls responded to per year. So sort of a signal that maybe this is more about firefighting than environment. The other thing that was interesting in the survey data was a strong signal around education, more years of education being associated with each of these 3 PFAS, and we're really not sure what's going on here. Maybe it is that higher levels of education get you into jobs in the volunteer fire service where you have more exposure, or maybe it's that more disposable income gets you more products that are PFAS treated. We just don't know. Then we also looked, we had 11 new recruits in this group. So we compared the levels among new recruits to incumbent firefighters. And we found that this PFDOA in this one department, again, was higher in incumbents versus new recruits, sort of another signal that maybe this is related to firefighting. Finally, I talked a few minutes ago about having begun enrollment of additional firefighters. So again, we have from our new department B, we have 33, and Department C, we have 11 firefighters. And so we compared levels of PFAS, again, very small numbers, very preliminary, but we see similar levels of PFNA and PFDEA and higher levels of PFDOA. So we're starting to think that there may be something unique in this one department about PFDOA exposure. And I'll tell you that this town has a history of pollution from the dye industry. And so that may be what's pushing this. But again, we do not know. So limitations, of course, of cross-sectional study. We don't have precise measures of firefighting exposure, but we do think that we know that this is the first largest study of volunteer firefighters, and we're hoping that we're framing ways to look at PFAS exposure in volunteer firefighters. We're hoping we are going to be expanding enrollment. We have funding from FEMA to enroll 300 more firefighters in New Jersey, and then around the country an additional 300. And then we hope to seek funding to assess maybe non-community comparisons of PFAS exposure, and then and levels and sources of exposure directly. So we have this fantastic collaborators and team, and thank you to our funders, FEMA and the Robert Wood Johnson Barnabas Health System and Cancer Institute of New Jersey. And we're happy to take your questions.
Thank you very much. We're going to hold questions until the end when we do the facilitated discussion. So if I could ask everyone to just write down your questions and keep that in mind. I want to move on to our next speaker. And thank you, Judith, for being well within the time. We greatly appreciate that. That will free up a lot more discussion at the end of this. We're going to transition to our next speaker. You can see the slides. This is Jody Wireman from the Department of Defense, Force Readiness and Health Assurance. So Jody, please go ahead.
Hello, good morning and good afternoon. And thanks to the NORA Public Safety Sector Council for conceiving of and moving forward on this PFAS meeting. I'm Jody Wireman, and I will be providing an overview of the PFAS blood testing effort for the Department of Defense firefighters. Slide 2. Like other attendees for this meeting, the DoD is interested in potential PFAS exposures experienced by firefighters and other occupations from their historical and ongoing use of PFAS-containing substances. We're also interested in better understanding what serum PFAS levels could indicate relative to health impacts to firefighters and others. That said, it was Congress who directed the DoD to offer blood testing to its firefighters. More specifically, Section 707 of the National Defense Authorization Act of fiscal year 2020, titled Provision of Blood Testing for Firefighters of the DoD to Determine Exposure to PFAS. In Section 707, it requires the Secretary of Defense, beginning on October 1st, 2020, to provide blood testing and document potential exposure to PFAS for each DoD firefighter during their annual exam, and it defined a DoD firefighter as someone whose primary job or military occupational specialty is that of a firefighter. Slide 3. With those requirements, the DoD developed a way ahead that included a number of activities that needed to occur before sampling could begin in October last year. The first part was to identify the firefighters to include both DoD civilian and military members. And when I say military members, I'm speaking of active duty, National Guard, and reservists who are firefighters. The services, Army, Navy, Air Force, and Marines, and the Defense Logistics Agency identified their current firefighters, those who receive annual physicals. There were approximately 20,000 firefighters identified who work at over 400 different locations. So you can see that this was quite a large undertaking with a relatively short period of time to get it implemented. In identifying a laboratory for serum PFAS analyses, the laboratory selection, we informally consulted the CDC on their analytical protocol and to get some insights on laboratory capabilities, how we would go about doing that. I think if we had a few hundred samples, the CDC, National Center for Environmental Health, may have been able to support us, but 20,000 sample was way beyond their capacity. The other challenge, I guess you could say, that we had was, or consideration was in choosing a laboratory. We needed a laboratory that conducted clinical analyses, and not all laboratories that analyze serum do so in a clinical fashion. And this was because Congress directed that the individual firefighter results are documented. That said, one laboratory met our requirements and was also associated with a DoD laboratory contract with military hospitals and clinics, so it made it that much easier to move forward with that. The PFAS panel that is currently being analyzed is PFHXS, PFNA, PFAS, PFOA, and it also includes PFBS and PFHPA. Next slide, slide 4. In addition to identifying firefighters and a laboratory, the DoD also needed to formally lay out the DoD firefighter serum PFAS testing requirement so that it could and would be accomplished with firefighters at those 400 locations. This was accomplished through the development of a number of products, which were shared with our interagency partners for awareness and input, and formally reviewed within the DoD to include unions as far as finalizing these documents. So the first document, the policy memorandum, it directs the DoD components, the services, the defense logistic agencies, those who actually, the firefighters report to them. It requires that they offer serum PFAS testing in conjunction with each firefighter's annual occupational medical exam. The memo also included 2 fact sheets, one for firefighters and another for healthcare providers. So, it was a challenge, a chain of command kind of thing, but that policy allowed the testing to be initiated. And as that policy memorandum was being finalized, DoD websites were set up to share the information with the medical community and employees. So the health.mil website that you see there is actually available to the public. You go to it now and find these documents. One of the documents, I think it's pretty integral to this effort, was a DoD guide that was given to MTFs, hospitals, and clinics. And it was really kind of a how-to document that walks providers through the process of sharing the fact sheets with the firefighters, offering the testing, conducting the blood draw, ordering the PFAS panel, and providing the results to each prospective firefighter. The website also includes the policy memo described above, a PFAS blood test results memo that results can be attached to provide to each individual firefighter, and a declination memo if the firefighter elected not to have their blood drawn or tested. And finally, the last slide that I have, slide 5. So like others, there have been some challenges in implementing the serum PFAS firefighting sampling effort due to COVID-19. But my understanding is that all DoD firefighters will be offered a blood test by the end of the fiscal year, 30 September. And blood testing of DoD firefighters for PFAS will continue annually for the foreseeable future. So if they declined over this year, they could be tested again next year, or they could have their second test next year. Analytical results for the firefighter testing are obviously of interest to us firefighters and others. So we're in the process of extracting DoD firefighter serum PFAS data from the firefighters' electronic health records where they're being maintained. This is being done by DoD epidemiologists who de-identify the data so it's not associated with individuals. And from that data, we'll be developing population level summary statistics, central tendencies for the firefighter serum PFAS data. So more to follow on the results. And then from a research study perspective, certainly the reason that we're here and others from the DoD on this, be able to attend this meeting is to find out what's being done within universities, firefighting community or others relative to PFAS. And I also wanted to indicate, or I also wanted to let folks know that a study that was funded a few years ago by the DoD, where the National Cancer Institute is going to be the primary investigator, is going to make use of the DoD serum repository, which has blood samples dating back to the 90s. It was originally taken in conjunction with HIV testing, but that blood serum is available for research if it's deemed necessary or applicable to the military. So this DoD serum repository is going to be used to look at Air Force members to evaluate whether there's an association between serum PFAS levels and testicular cancer. So that effort is just getting started, and we'll see where that goes. So that ends my presentation. Thank you.
Thank you very much. We do want to hold questions to the end. I do see someone has their hand up. Andrea, is that just a question that we'll reserve for the facilitated discussion, or are you having potentially some technical issues?
Oh, no, thank you. Sorry. End of discussion is fine. No technical issues. Thanks.
Perfect. Thank you. Okay. Thank you very much for your presentation, Jody. And we continue to be ahead of schedule, which is wonderful. Next speaker will be Jackie Goodrich from the University of Michigan. Jackie, go ahead.
Yes, I'd like to echo the other speakers' sentiments. Thanks for organizing this and for having us here today. I'm going to build upon what Dr. Burgess was just talking about and move away from just measuring the exposures and talk about some of the biomarkers of effect that we're starting to measure in the same cohort. So to make sure we're all on the same page about what epigenetics is, I think of epigenetics, literally it means on top of the genome, but we think of it biologically as being changes in gene function or expression that occur without a change in the sequence of the nuclear DNA. And there's many types of epigenetic regulation. DNA methylation is the one I'll be talking about today, but there are also non-coding RNAs and histone tail modifications and other things that impact chromatin accessibility. As far as DNA methylation is concerned, it's actually a pretty simple modification to your DNA. All it is a methyl group addition to a cytosine, which is one of your 4 DNA bases. But when you have a lot of these in especially the promoter region of a gene, it can block binding of transcription factors and inhibit transcription, which is its general function. And in mammals, it occurs at what we call CPG sites, which are cytosines next to guanines. So you may hear me refer to that. So why do we care about this in the context of PFAS and studies of firefighters and things like that? Well, we know from lots and lots of studies that various environmental factors can impact the epigenome, including DNA methylation. And so these exposures that we know impact the epigenome include metals and air pollution and things in diet and even things like stress. And there's some studies now that show that PFAS may also modify epigenetic signals. And this is important because it's not just a one-time change that has a limited short-term effect. When an epigenetic modification occurs, the next time that cell divides into a new cell, the change goes along with it and is replicated in the new cell. So these effects can be quite stable and can have long-term effects ultimately for health and disease. And because of this, we wanted to start looking at whether we can measure epigenetic biomarkers in firefighters and whether they would correlate with exposures, especially to PFAS, that we've measured in the same firefighters. So to get at that, I'll tell you about two of the questions we asked in this domain and what we've seen so far. The first question, we looked at something about called epigenetic age, which I'll describe to you in a second. And we sought to address the question of, are serum PFAS concentrations among structural firefighters associated with accelerated epigenetic age? And the firefighters that are included in the parts of this, both studies I'll talk about from here, are part of the FFCCS and another study that Jeff did before that. They're all active structural career firefighters that had exposure analysis for PFAS and also DNA methylation analysis done. They're from 3 departments and they're primarily male and white non-Hispanic. So what is this epigenetic age that I'm going to tell you about? So this is something that's really cool. Actually, there have been a lot of researchers that have figured out DNA methylation at certain CPG sites, certain genes, is really, really highly predictive of chronological age. And if you kind of make a signal of a bunch of these sites combined, then you can get an estimate of someone's biological age. So they found that your epigenetic age often correlates strongly with your chronological age, but if you've had various exposures or various detrimental health conditions, you have this accelerated biological aging that's reflected in this measure. And the figure here shows 5 different clocks that are similar, but calculated in slightly different ways and have been very well published. And these, what's neat about these is that not only have they been shown to be associated with different kinds of exposures, if you have this accelerated age from measured by these clocks, that's often correlated with cancer, higher mortality, higher cardiovascular diseases, and lots of other bad things, basically. But what's not known is if PFAS is one of the exposures that modifies this epigenetic age. And similar to what Dr. Burgess talked about, we measure 9 PFAS analytes. Most of them were detected in the population. 3 of them weren't that widely detected, but the rest were, and these are all legacy compounds here. And so here's a summary of some of the results that I saw. Going across each column is one measure of epigenetic age, and each row is one of the PFAS that we measured. And in models adjusting for various confounders, potential confounders in this relationship, we saw that 3 of the PFAS, BFHXS, SMPFOS, and NPFOA, were significantly associated with accelerated age, so that's what's considered bad, in several of the clocks. And the bolded estimates are the ones that are statistically significant. And then you see that some of the other PFAS were trending in this direction too, but they weren't significant. And then the longer change PFAS weren't really associated with the clocks, except for 2 of them were inversely associated with 1 clock. And so I'm not quite sure what that means yet. So moving on to beyond age to what specific genes might have changed because of PFAS exposure, we looked to see if these PFAS concentrations were associated with certain kinds of genes and what pathways those genes might be in. And this is a very simplified version of the results, but basically we had methylation data at over 700,000 different sites throughout the whole genome that represents all the genes that you have and multiple sites within each gene. And when you do analyses with that much data, you have to really make really strict criteria for significance calling because there could be a lot of false positives otherwise. So what ends up happening is sometimes you might not be picking up all the truly, true biologically significant results. But here, the strongest signals that we did get showed a couple of genes that were associated with PFOA and PFAS, and then also with some of the longer chain PFNA and PFDEA, as well as PFOA, had genes associated with them as well. And when it came down to what these genes were, when we really dug into what they were and the pathways they were in, quite a few of them were involved in processes related to cancer and carcinogenesis. Some of them have been published previously as biomarkers of certain cancers. And then some of them are also involved in immune function. And we know that certain cancers and immunotoxicity are two of the potential effects that PFAS exposures have. So this was really interesting since we're screening this in healthy firefighters. So as a quick summary, among structural firefighters, we saw some evidence that there were several PFAS that seemed to be associated with accelerated epigenetic aging. And then there were also several PFAS that are associated with methylation patterns at certain genes. And these are things that we're excited to keep pursuing. And we're doing epigenetic analyses now in more firefighters, including ARFF firefighters who might have higher PFAS exposures, and also other groups of firefighters that weren't necessarily selected for studies of PFAS, but which we have PFAS data on as well. So we'll see if some of these markers hold up when we screen them in more people. But with the limitation at the moment that these associations, we can't say that they prove causality right now. And we also don't know right now if the exposures were from the occupation or from the surrounding environment, but there are people on the research team that are looking into that more, including the kinds of things Dr. Graber was talking about. And just one more quick plug for why we use epigenetic biomarkers. They could be really useful in occupational risk assessment to, kind of assess the effects of toxic exposures before you see some, overt health outcome like a cancer. They may also be able to help us understand mechanisms of toxicity and be developed into screening tools that could predict, what individuals might need more prevention or intervention strategies because they're at risk for disease later on. And I'd like to thank the same great team and funding sources that Dr. Burgess mentioned. We actually have even more people than this that work on the project. And thanks again for having me here today.
Thank you very much, Jackie. We appreciate the presentation. We're going to go ahead and move on to our next speaker, and that's Carrie McDonough from Stony Brook University. Carrie, go ahead.
Thank you. Thank you so much for having me today. Better. All right. Yeah, so thank you so much for having me. I'm very excited to be here today to talk about exposure to PFASs and specifically expanding kind of our lens to consider exposure to novel compounds and compounds that aren't typically included in biomonitoring studies. So I wanted to start out really quickly. Everyone who's in this meeting clearly is familiar with the fact that PFASs are found in all of our bodies. I just wanted to highlight, that really early on in the 60s, scientists were analyzing their own blood and finding, unknown, unidentified fluoride. And, how far we've come from here, from these first suggestions in the 70s that, there's this widespread contamination of humans with organic fluorocompounds. And I bring this up mostly to highlight the fact that we are only as good as our analytical chemistry. And as our analytical chemistry continues to advance, our understanding of what we're exposed to and also the questions that raises are going to keep advancing as well. So with that in mind, nowadays, with the analytical chemistry tools that we have, we've discovered that there are thousands of PFASs. This is a figure from 2017 that, is already out of date. It says over 3,000. We're up to, I think the most recent figure is something like more than 8,000 compounds that have been identified with a wide range of different structures and physicochemical properties. So with that, of course, it raises the question then, if there's all these novel PFASs, are they bioaccumulating and are they in our bodies along with the ones that we're more familiar with? And there's been some studies over time into this. This is an early study showing basically percent composition in residents of Chinese cities. The bars in color are target analytes that were identified, and the black is unidentified organic fluorine. And you can see that there's a fair amount that seems to have some kind of geographical trends to it of unidentified fluorine. More recently, here's a study from 2020 where researchers measured a Swedish cohort which had been drinking HFLF impacted water and found that over time, the percentage of extractable organic fluorine that's accounted for by target PFASs is decreasing, which is implying that the amount of the PFAS in our bodies that we don't understand may be increasing over time. So some early indication of the bioaccumulation of novel PFASs actually came from early studies in 2015 on occupational exposures among firefighters. So this is one of the first papers to find novel PFASs in human blood. And here you're seeing, the PFAS that we're pretty familiar with, things like PFOS and HXS. And then at lower levels, all of these other compounds, pentane sulfonate, as well as chlorinated and ketone substituted compounds. And they found that these compounds were present at higher levels in firefighters than in a control group of office workers. So how does all this then lead into what I'm working on now? So I have been collaborating with Professor Jeff Burgess and his group at University of Arizona to analyze kind of a subset of samples, paired firefighter serum samples, comparing baseline and post-fire, so samples that are from a chronic annual exposure assessment, and then other samples that were taken directly after fire events. And the question we want to answer here is, how do exposures that are related to these structure fires impact the post-fire serum PFAS profile? And to do this, we want to analyze the serum for an expanded analyte list, including using non-target strategies to really start getting at not just these compounds that we regularly monitor, but also, what else might be present in these samples. So this is something I've been working on with my student, David Dukes at Stony Brook. So the first thing we've been working on is developing a comprehensive method for PFAS serum analysis. We want something that's highly sensitive and PFAS selective, but also inclusive of a lot of different PFAS. As you saw, there's so many different ones to try and capture. So for that reason, we've been developing a new sample preparation method in which we acidify our sample for protein denaturation in a cartridge and do a protein crash followed by a lipid removal with a cartridge that basically works by hydrophobicity and size exclusion. So it's not very selective for a specific ion or anything like that. So that's a kind of one-step preparation followed by transfer to an autosampler vial, and then we run on liquid chromatography coupled with quadrupole time of flight mass specs. This is high resolution mass spectrometry. So just to show you a little bit of data on kind of the performance of the method so far, here's the quantitative validation using spiked serum samples. These are spiked bovine calf serum samples. And here we spiked with 40 different PFASs that we're actually targeting and quantifying. And we're comparing the concentration that was measured to the known concentration in the spiked sample. And we were pretty happy with these results. Everything is pretty much within the 30% dotted lines. And you can see we've got a pretty expanded, nice analyte list here beyond kind of what's regularly monitored in something like NHANES. Our detection limits are pretty good. 17 of these compounds have detection limits below 0.1 nanograms per mil, and the rest kind of are, we have a range that we're still working on getting some of these down. So here's a summary of the target analyte concentrations, and I apologize for how many colors. When you have this many compounds, you start to kind of run out of colors. I tried to simplify by kind of showing the sulfonates in red, and then the carboxylates in blue, and then all of the other compounds are in green. And the main takeaway here, when you look at the concentrations in each of these samples, they're divided into the post-fire and the baseline samples. It's dominated by the sulfonates that are targeted in these samples. And mostly the hexane sulfonate branched and linear octane sulfonate, and then PFOA. And comparing to NHANES, we see with our medians compared to adults from NHANES from 2017 to 2018. This is for the frequently detected compounds here. And in general, our medians are pretty similar. I would take this with a bit of a grain of salt because it's such a small number of samples, but they are pretty close for most of the compounds, with the exception of branched PFAS, which seems to be elevated in our cohort, and hexane sulfonate, which seems to be elevated. So some other takeaways from these samples so far. This is all very preliminary, and we've only run half the samples, so we are going to do additional analyses on more of these samples. So far, we are not seeing pronounced differences that are readily observable between the two groups, the post-fire and the baseline, but we are going to keep inspecting that. We did find that some compounds were detected more frequently in the post-fire group, but these compounds have pretty low detection. They're mostly the short chain sulfonates, which would make sense, as well as the short chain sulfonamides. And I also wanted to highlight that pentane sulfonate and this cyclic compound, perfluoroethyl cyclohexane sulfonate, are rarely included in monitoring lists, but they were detected in more than 90% of individuals in this study from both groups with very low concentrations. So we have some kind of chronic low-level exposures going on that aren't regularly being monitored. So our next steps as we continue with the study are to start on more non-target screening approaches. So during the quantitative analysis, data was also collected on peak intensity for all masses between 100 and 1200 Daltons. So this is a huge amount of data for both current and retrospective screening that we can use. And we've been working on top-down suspect screenings. This is when we look for compounds that we suspect might be there using extracted ion chromatogram lists of thousands of PFASs. And I'm also going to be doing bottom-up non-target screening. So that's where we would compare groups, like our post and our baseline groups, look for compounds of interest, and then focus in on those compounds and try to figure out what are these structures that we haven't identified. So some very early preliminary suspect screening analysis data. We found about 171 peaks of interest after blank filtering, which we're now working on manually checking and finding standards for these compounds to confirm their identities. Here's an example of one that we're fairly confident in. This is a ketone substituted perfluorooctane sulfonate that was found in that previous study I mentioned from 2015 of firefighters as well. We had a tentative identification of this in more than 90% of our samples, and the relative peak areas suggest low levels, probably under 0.2 nanograms per mil. So with that, I want to thank everyone who's been collaborating with me on this project. It's a really exciting new collaboration for me. And yeah, thank you all for listening.
Thank you, Carrie. Great presentation. We have one final speaker in this section, and then we're going to go to the discussion. So our final speaker in this session is Miriam Calkins from CDC NIOSH, and she's with the Division of Field Studies and Engineering. Go ahead, Miriam.
Well, I'll start talking while that comes up so we're not wasting time. But I do want to thank you for the opportunity to present here. And I'm going to switch gears a little bit at the end of this segment and talk about a feasibility study for exposure and health indicators in other occupational populations. So moving a little bit away from firefighters and also talking about a study that we are about to get into. So this will not be nearly as results heavy as the previous presenters, which were all really very fascinating. So I'm presenting today as a research industrial hygienist, and I'll be presenting with Jen Roberts, who's a chemist in the Health Effects Laboratory Division. Next slide. So when we talk about occupational exposures, I think it's important to take a step back and think about the life cycle of the product or the chemical as a whole. So entirely, you can click. I'm so sorry about this. We want to make sure that we're including consideration for workers who may work in the manufacturing and integration of chemicals and products, the use of those products, the disposal and release of those products, and then additionally, anyone who may be involved with any remediation of potential contamination or disposal sites. So with firefighters, I think that that exposure typically occurs kind of in the middle of that life cycle with the use of products. When we talk about the use of AFFF or possibly turnout gear, and the populations that we're really interested in for this study are earlier on in the manufacturing of chemical compounds and the integration of those compounds into products. There's often industry, occupation, and task-specific differences that are really important to consider when we dive down this route of looking at other work settings. But they can be very informative to look across populations as well as understand some of the differences within those as well. So for, since we're talking about firefighters today, some of the industries that we're interested in that are somewhat tangential but related are industries such as textile manufacturing, and then also industries where there may be substantial exposure through routes that are not typically assessed in the environmental route, such as inhalation or potentially dermal exposure as well. Tyler, I think you have to click twice for the next one. Go ahead, click again. Thank you. So just to give you an idea of how the literature thus far has illustrated occupational exposures primarily, here's a figure of PFOS, and this is in serum, plasma, or whole blood, although it has all been, I believe, compared onto so that it's appropriate for serum across the board. So the literature, and this is slightly outdated, I think there's one or two newer studies that should be included at this point, but the literature thus far has really focused on 3 occupational populations. It's focused on firefighters, and they're in red, ski wax technicians, and they're in orange, and then PFAS chemical plant workers in purple. And I have compared these populations to some studies of non-occupational populations. They're in blue. Keep in mind that the data presented, it does cross a number of years, over a couple of decades, and we have seen that PFAS has decreased in the general population over time. So that's just something to keep in mind. And then also the x-axis is on a log scale. So these exposures are actually much farther apart than they may appear on this figure. But you can see that there's groupings by industry. Typically the chemical plant workers tend to be on that higher end of exposure. And then there's some clustering, but also some overlap with the firefighters, the ski wax technicians, and then the general public. Some of the higher blue dots, those general public dots, are actually populations that live near a point source. So they do have a known source of local contamination and would not be what you might consider to be a typical background for the US. So when we dive into an understanding of how workers are exposed, we have these groupings by industry, but then also clearly some differences within each group, within firefighters, within chemical plant workers, and we need to understand how the tasks and the specific exposures within those occupations may impact exposure. Next slide. So for this study, we have a few objectives, but I'm really going to focus on the last 2 that are listed here. That is to conduct an exposure assessment focusing on current ongoing exposures in these populations, and then also to conduct an evaluation of a limited panel of health indicators that we can measure in the workers. We are plan to enroll 150 workers, and these would be workers from some of those, as I already mentioned, those earlier life cycle industries that we anticipate to have some of the higher or more moderate use of PFAS. These primarily will include manufacturing and services sectors. Next slide. So this is a cross-sectional study. And for the exposure, we are including biological samples. Primarily, this will include serum. Full shift breathing zone air samples, and Jen will talk a little bit about those methods here in a few minutes. And then also including observations of job tasks, personal exposure equipment, and all of that information that may be really important to understanding how that person's exposed. For the health assessment, we are including some survey questions, self-reported diagnoses, information such as that, and then also analyses of indicators for cardiovascular health, kidney function, thyroid function, liver function, and immune response that we can measure in the serum of the workers. Next slide. So there's some interesting things that we need to consider, and many of my colleagues have already alluded to some of the complexities of measuring human exposure for PFAS. So there's a few different ways that you can measure or try to evaluate exposure in an individual. Biomonitoring is probably the most commonly used for PFAS, primarily serum, but you can also use other matrices such as urine. However, there are some limitations that we can discuss later on or may actually come up in other presentations later today. There is measurements of PFAS in the environment. Air is one that we're including in this study, but other studies have included dust, textiles, and so on. And can really give you an idea of what's in the environment, but may not actually be fully indicative of what the person has in their body. And then lastly, there's some modeling that can be conducted using work history and some measured exposures either in that population or in other populations to try and evaluate what somebody maybe you don't have information on might have in terms of their exposure. For PFAS specifically from a measurement perspective, typically there's this targeted list of compounds. A number of us have alluded to using the panel developed by NCEH for NHANES, and that is what we do plan to use for this study. But there are obviously a lot of efforts going towards expanding that panel or using other approaches such as non-targeted or semi-targeted analyses. This represents a very small fraction of the total PFAS that are out there, as was illustrated really very nicely by a previous presenter. And it may or may not actually include information about precursor compounds. It also is developed for a certain population, and workers may not be that appropriate population. Can you click once, Tyler? So there's, I think that's really what I was trying to highlight there with the targeted list. And then can you click one more time? So there's also this difference between active ingredients and contaminants when we're trying to figure out what somebody might be exposed to. So we may have an understanding that either these terminal compounds at the end are the active ingredient that's integrated into a product. But when we're diving into an evaluation, there are probably, with PFAS in particular, a lot of other contaminants that are introduced in the process of either making that chemical compound or during the process, things that have transformed from earlier forms of the PFAS to these other terminal ends. Lastly, I do want to mention the time between exposure and the test is really vital. I think a lot of us are probably familiar with how there are some differences in the half-life of PFAS, and a lot of those are related to the length of the carbon chain. But that can be really vital when we're trying to figure out exactly when to measure the exposure in an individual in relation to the potential exposure and when you have access to the participant. So can you go to the next slide, please? Jen? Jen, I think you're muted if you're talking.
Thanks, Miriam. Can you hear me now? As Miriam alluded to, we are developing an air sampling and analytical method for PFAS through our contract laboratories actually doing the majority of the work. And the method is going to be a targeted method with 12, what we're calling the short chain PFAS compounds in both their acid and their base form. And the compounds that we're including at this point are the C4 through C8 carboxylic and sulfonic acids, as well as HFPODA and Adana, which contain the ether groups. For this air sampling method development, we're using the OSHA versatile sampler, more commonly known as the OVS tube. For those of you not familiar, the OVS sampler contains both a filter and sorbent beds to collect both particulate and vapor phase. And then on the analysis side, we're using HPLC MSMS. Next slide, please. And then I just wanted to give you a brief rundown of where we are in the development. We're following the NIOSH guidelines for air sampling and method development and evaluation. And the major steps in this process are listed here in this table. We've completed the basic measurement steps, and the method limits of detection and quantitation is noted by the check marks. And then we are working on the STARD studies, the recovery and air sampling studies. These studies include evaluation on sample breakthrough and the effects of humidity in the environment. And we do this at multiple concentrations and multiple humidity levels. And then after these studies are completed, we'll complete the 30-day storage stability study. And then, as Miriam mentioned, this work is going to support the field work she's undertaking for the feasibility study. And it'll eventually be included in the NIOSH Manual of Analytical Methods. The method development and MAM process take a long time to complete. In many instances, 2 to 3 years to get into the NMAM. But once we get these evaluation steps completed, we'd like to compare our method with methods from other agencies. And also in the future, we'd like to expand the targeted compound list. But each added compound would have to undergo this kind of rigorous evaluation we do. So those will take some time to add them in. Miriam, I'll turn it back to you.
And Tyler, you can go to the next slide. Okay. Last slide, and you can click 2 more times, I think, and we'll just have all, yep, good. So before I close out, I just wanted to mention some bigger picture issues that we consider whenever we evaluate occupational exposures, especially for exposures that there's a lot of concern about that we don't fully understand in terms of the exposure in different populations and guidance for workers. So there's clearly for PFAS in particular, there's a lot of limitations of the existing research. There are likely mixtures that we are not capturing, specific compounds that we're not capturing in our analytical methods. A lot of the existing literature has also focused on routes of exposure, particularly the health literature is focused on routes of exposure that are really important for a lot of the general population, primarily that oral route of exposure and ingestion, that is, but may not be as relevant for some workers, including firefighters. There's also been this transition towards shorter chain PFAS, where there's maybe there's less information about that in terms of the toxicological information, and then also some of the exposure information, and clearly limitations of analytical methods. There's also, I think, some really important considerations when we talk about what to do about these exposures once we've measured them and we're trying to figure out how to approach either an intervention or how we're going to deal with reducing exposures for the workers. And I just wanted to include a couple examples. So there's some safety considerations, and for example, in the context of firefighters, replacing the foams where we know that there may be PFAS in them with something that there is no PFAS. We want to make sure that there's the same efficiency. So we're not trading the risk of PFAS for the risk of having an agent that is not as efficient and not going to put that fire out. So making sure that we have something that is appropriate as a replacement so that we are not just trading one risk for another. Similarly, shifting populations that are at risk, taking a bigger picture back. So when we start to remove and dispose of materials, we want to make sure we understand how to do that in a way that's going to protect the workers that are then going to be dealing with the disposed products, so that we aren't just shifting the risk from one population that's using a product to the population that is now remediating an environment or dealing with the disposed product. So making sure that we're protecting all populations that are coming in contact with it. And then lastly, that there's a lot of limited, not a lot, there is very limited occupational reference limits and guidance for people to reference and to look at and to include in training materials and to integrate into their safety programs. So trying to provide that information as we move forward. So I think I'm probably over on time, so I'm going to stop right there. Thank you.
Thank you, Miriam. Thanks you, Jen. We have 10 minutes left for our facilitated discussion. Originally, I had seen a hand up from Andrea, but I actually don't see her in the meeting at the moment as I'm quickly looking through. So perhaps she had to drop off for a second. If you are still in the meeting, Andrea, please raise your hand again, and then we'll know you still have a question. Just so what we're going to do here, I'm going to share my screen just to remind everybody of the speakers' names and their titles of the presentations, just to help if you have a specific presentation. Can you confirm that you can see my screen?
Yep.
Yes.
Perfect, because it's not giving me the nice little highlight it should be. Okay, so here you can see all the names of the different speakers. I'd like to open it up. You can use the raise hand feature, you can use the chat button feature, or you can just go off. I see that Sue Fenton, you have your hand up. I'll go to you first.
Oh, thanks. I actually, this is really for the whole group. I mean, I know everybody was very specific to make a point that they were looking at legacy PFAS. But, you know, with the contemporary AFFF use, have there been any studies started looking at exposure to some of the fluorotelomere-based C6 and smaller PFAS in firefighters, especially those that would use AFFF?
Open that up to our panelist speakers.
if someone else has a better answer, please stop me, but I'll try. We have been collecting samples and storing them within the firefighter cancer cohort study for additional analyses as opportunities come up, and we have the ability to look for compounds. So as you saw with Carrie's study, what she was looking at initially were kind of the exposures associated with a fire and wearing gear, obviously, because you're going into the fire with gear as well. We could also look at differences between, let's say, airport firefighters who have a history of exposure to AFFF and municipal firefighters. So, I mean, that is an opportunity, but we don't have those things identified yet. Also, Miriam was, one of the things that she was going to do as part of the project was to actually look at AFFF use during training. Probably, thankfully, most places aren't using AFFF in training anymore because that's reduced exposure. But if opportunities do come up, that we could collect that information as well.
And Jody, do you guys have a chance to incorporate some of those newer, smaller compounds into your list?
No, we haven't been able to do that as yet, and I think to the work that maybe Miriam Calkins was hoping to do was I think part of that effort related to timing of when that blood collection would have to occur after, say, AFFF was used to gain some insights on that type of exposure before it was eliminated. Over.
Yeah, because I saw that Carrie has some of the chemicals that I'm thinking about on her list. And I just didn't know if, because it does sound like several of you are working together, if maybe some adoption of Carrie's methods might help.
Yep. So we do plan to send some of those samples to Carrie. So she will be, and then we'll be comparing that with what NCEH reports in those samples. So that is part of the plan.
Great. Kenny, I see your hands up. Do you want to go next?
Sure. I think it's kind of a piggyback question to Sue's question, and maybe more for Jody. I'm curious with what the DoD is doing, because the DoD, you have airport rescue firefighters, but you may also have more, traditional structure kind of firefighters. And I'm wondering if there are plans to kind of do that comparison among the DoD data that you're collecting. And then related to that also is, are there any plans to kind of look at the environmental exposures compared to the potential occupational exposures?
Yeah, we really haven't gotten into the analytical piece as yet, and have been considering formulating a questionnaire, but there's probably also opportunities to allow us to get that information from the units themselves if we drill down into that level. So I think that opportunity could exist in the future for the DoD. That's just my personal opinion, but I think that the data could be there, and if this becomes an annual requirement, I think we can improve upon it and collect additional information as we move forward. Thanks.
Thanks, Jody. We do have 3 different questions that are in the chat, so I'm going to take them in the order we received. The first question is from Samuel. This is a question for the new detection method being developed. Will this method be developed for in the field measurements and detection, or would it be benchtop method?
Hi, this is Jen. I think that for the air sampling method development, that's going to be a benchtop laboratory method.
Great, thank you, Jen. And Samuel, if you have any additional follow-up, you can always put it in the chat. I'll go now to Celeste has a question. And oops, sorry, my chat is updating. So move. There we go. Is there any data on firefighters' PFAS levels in their bodies after they retire? Do the levels go down in a person's body after they have been exposed? Is there anyone aware of any data about that or have perspectives?
I'm not aware of any. Realize that these all have elimination half-lives, so they will go down. The question, but also realize that those are elimination half-lives. They don't take into account continued exposure. So you have things that you're continuing to be exposed to, the levels won't go down as much. But we have, as you saw, when we were looking longitudinally in our group, these levels tended to go down for almost all of the legacy chemicals.
I'll add to that and say that there have been studies, I believe it was the Rotander study, the Australian study, where they evaluated PFAS in serum before and after a major change in training practices, where AFFF was eliminated from their training. And they did see that with that change in practice that PFAS did go down over time. So don't quote me on the study I just cited. I think that's the one it was, but whoever that was, if you reach out to me, I can get you the study.
So Celeste, if you want to just touch base with Miriam afterwards. And we have 3 questions left in the chat. Let's go ahead and answer them, and then we'll take our break. If anyone has additional questions, they can pose them to the different speakers of the group through the chat. Continually, people can monitor it and provide answers there. But these 3 questions we'll ask verbally. The first one from James Smith: are analytical methods for PFAS comparable? Several presenters compared their data to NHANES. What was done to ensure compatibility?
Again, I'll jump on that really quickly. We had the same laboratory, NCH, analyze our samples to date. Judith can talk about, the New Jersey laboratory, which has comparable methods. Also, when we select our comparison group, we consider people that are of similar age, and also realize that the NHANES tends to oversample for minorities so they can get a large enough and statistically valid group. And so you need to take that into account when you're also selecting identifying your comparisons.
Well, similarly, our comparisons were among white, non-Hispanic white males in the same age group, but then we use the New Jersey Department of Health testing laboratories, and they are trained by CDC to conduct PFAS testing, and they do regular quality control together. So they are considered comparable.
Thank you, Judith. Thank you, Jeff. We're going to take these last, the 2 questions we had, and then there's a 3rd question that hopefully folks can answer through the chat. The next 2 questions are for Dr. Wireman. First one is, has there been a trend for military and civilian firefighters to opt out of blood testing? So that's the first question, and I'll read the second question when you're done answering that.
Yeah, the quick answer to the declination rate is we don't have that information yet on whether there's a trend at all. And go ahead and read the next question.
Could you clarify if, in addition to current DoD firefighter occupational monitoring, are veterans concerned with their past DoD firefighting exposures able to obtain PFAS blood testing via VA primary care in consultation with their physician?
Right, and I think, there's an interest, potentially for DoD firefighters who are no longer doing firefighter work and not doing annual testing. So the testing that the DoD is doing right now are for basically current firefighters or those who receive annual tests. I know that the Veterans Administration has been asked about veterans and whether they could get the test. And well, if you go to their website, and we work with them all the time, they're not at this point offering tests to veteran firefighters, at this point. So the only DoD veteran effort relates to current firefighters. And I wanted to kind of go on with Jim Smith's question. I think he was talking about new methodologies for new analytical methods that are being used, and how they could be compared to NHANES as well, not necessarily those who are using the exact methodologies that NCH is using relative to, for some sort of a comparison. Thank you.
Thank you. With that, we're going to give everyone unfortunately, we're down to just a 2 or 3 minute break to just comfort break here. I will draw your attention, experts. There's one remaining question in the chat. So we're not going to verbally answer it. We're just going to type back for Rick Davis. He said, data research on the transport of PFAS into the body at rest and active, anything specific to firefighters, what PFAS species were studied. So we're not going to verbally answer this in the interest of time, but please, experts, if you would respond back to Rick's question as your time permits today. Let's just take a few minutes, a break, comfort break. We will get started again at right around 2:25. So just enough to use the restroom, folks. Thank you. Okay, folks, hope that was long enough for you to take a break. I'm going to turn it over to my co-facilitator, Carol Brown. Carol?
Thanks, Susan. My name's Carol Brown. I'm the Deputy Director at the Center for Health, Work, and Environment at the Colorado School of Public Health, and I'm the co-chair of the Public Safety Sector Council. We're going to jump right into session 2, following a great first session. Our first speaker today is going to be Christopher Ray, the Associate Director for the Agency for Toxic Substances and Disease Registry. And it looks like you have your slides up and are ready to go.
And can you hear me?
Sure can.
Okay, great. Good afternoon, everyone. My name is Chris Ray, and it's truly my honor to be here to talk to you about the work that we are doing at ATSDR as it relates to PFAS. And I must say it's an honor to be here with the NIOSH colleagues. If you don't know, I started my career at NIOSH within HETAB conducting health hazard evaluations. So good to be involved. Also, I want to recognize Antonio Califate, who's in the meeting. Many of you collaborate with her from the laboratory side. She leads our PFAS analysis group within the NCEH Division of Laboratory Sciences. And also Rachel Rogers is on, and she is our PFAS science lead as part of our community of practice. So just a little bit, ATSDR is very similar in many ways to NIOSH. We both were created as part of major congressional legislation. NIOSH, as you know, the OSHA Act of 1970. ATSDR was created through the CERCLA and Superfund regulation, where EPA was set up to be the lead for regulating hazardous waste and everything around that. And ATSDR is the public health agency. And in the Superfund Acts, we were asked by Congress to work to identify human health effects associated with hazardous substances and to respond to emergencies. But really the most important part of our legislation is this mandate to work directly with communities, which makes working at ATSDR interesting, because in everything we do, we take a lens on how we can best work with the community, how we can help them understand what tend to be complex toxicologic and physiologic principles, and to answer some of the basic questions around are the exposures I'm receiving through the air or drinking water impacting my health. We work closely with our state, local, and tribal partners, and in everything we do, we think about risk and risk communication. So we look at PFAS exposure from a little bit of a different lens than, let's say, you would at NIOSH or some of these presentations, in that we do not, we typically focus on environmental exposures versus occupational and workplace exposures. Now, there's definitely things we can learn from each other. It was good to see Miriam Calkins. We partner very closely with her. And when we think about PFAS exposure, we think about how drinking water can become contaminated, how food sources can, how our food supply chain can become contaminated, and then also how exposures in the home may impact body burden. The other part of this is we're very engaged in looking at PFAS exposures during pregnancy. and especially while breastfeeding. And in thinking about what are the right policies around nursing mothers and continuing to breastfeed. So we started down this journey back in 1999 when NCH and ATSDR worked with NHANES to start measuring PFAS in the general US population. And so when we say things like that, 98% of the serum samples collected by NHANES has PFAS, or that the majority of people in this country tend to have some level of PFAS in their blood. All of this work started in 1999 and continues through today. We developed our first methods to look at PFAS in human serum and in milk in 2004. In 2007, we expanded these analytical methods to include 11 PFAS compounds, and now there are several PFAS compounds. And we also, at that time, started documenting the declining concentrations of PFOA and PFAS in human serum through the NHANES studies. In 2009, we released our first draft of our toxicologic profile for PFAS compounds. And for those of you who are engaged with this, you will have noticed that the most recent version of this toxicologic profile was published 2 weeks ago. And we are continuing down the process to update what we published 2 weeks ago. So, you know, thinking about, are the MRLs that were done in the most latest final version of the tox profile, does the data and scientific literature still support that? And are there additional PFAS compounds where we may now have enough toxicologic information or human health information to establish additional MRLs? Our first real large-scale biomonitoring investigation was in Decatur, Alabama in 2010, where we found elevated levels of PFAS in blood concentrations of people in the communities of Decatur, Alabama and the surrounding area. The source of this PFAS contamination was manufacturing facilities in that area. And that's probably where I got my first PFAS exposure, because I was born in Decatur and lived my first 8 years in that area. So 2012, EPA started requiring all community water systems serving 10,000 or more customers to start monitoring for PFAS twice a month. And of course, in 2013, the C8 panel came to their conclusions. In 2015, we once again updated our toxicologic profile. We added, ATSDR added in 2017 PFOA, PFAS, PFNA, and PFHXS to the substance priority list. And the work goes on to where we are today. And what I'm going to talk about today is the current work that we have at ATSDR to better understand how PFAS can impact human health. And we have a lot of different efforts on in this area, but most of this work centers on the three major efforts, which are the exposure assessments that that we are currently delivering on, where to understand PFAS concentrations in communities near current or former military bases, our PEAS health study to examine health effects from contaminated drinking water, and then building on the PEAS study, our multi-site health study that is just now starting to kick off. So currently, this is what our map looks like of the different types of efforts and studies that we have related to PFAS. We have 28 projects across the country. Some of these are community health investigations, either ATSDR funded or state led, or through a cooperative agreement between ATSDR and the state through our Apple Tree program. These sites also include our multi-site study sites, which there are 7 of those, our PFAS exposure assessment sites, and this is the work that I previously mentioned on looking at PFAS exposure and drinking water exposures in communities where the source is believed to be from a nearby current or former military base. And then also our ATSDR-led health study in peas. And at the end of the day, we're really looking at a couple of long-term objectives. And we like at ATSDR to think strategically about work like this, and that is to understand how and where and what degree of exposure is occurring in affected communities, to examine the relationship between PFAS exposure and health effects, and then to identify strategies to prevent or mitigate exposure. So the exposure assessments, as I mentioned, in 2018, Congress, through the NDAA, National Defense Authorization Act, tasked ATSDR with conducting no less than eight exposure assessments at former or current military sites with high levels of PFAS in the drinking water. In this study, we've enrolled both adults and children. And through this study, we're collecting both blood and urine samples. On the right, you will see the 10 sites where we're conducting the study. 2 of the sites were to test or pilot the exposure assessment protocol. Those were the Bucks in Montgomery County, Pennsylvania, and the West Hampton, New York sites. And we used what we call our PFAS exposure assessment technical tool, or the PEAT, which is our exposure assessment tool. And if you're interested in using it, you can go on the ATSDR website and download the tool. And we are almost finished with the data collection phase of our exposure assessments. I believe the last batch of results are being announced this week. And we have started working on the site-specific reports for each of the 8 sites. And then after that, we will, of course, publish a final overall summary report of that includes all 10 sites. It is important to note that currently almost all participants have received their results of their PFAS exposures. And if you're interested in getting some summary information as to what the exposures are for the sites where the data has been released, you can go to the ATSDR website and you can navigate to a summary page. For instance, for Lubbock County, Texas, you can navigate to a summary page that will have summary data for the Lubbock County community. I got a little bit ahead of myself, but here's the process we're using. And as you see, we're almost finished, and we're at the stage where we're starting to write final reports. And next steps, we're going to continue to evaluate the data. We're going to be writing final reports with findings for each site and one overall final report. And we're also hosting information sessions to talk with community members. And through our PACES, or Pediatric Environmental Health Specialty Units, we're doing work to educate physicians in these communities on what to expect if the people from the community come to them with questions about their PFAS data. The multi-site study, is a large national study to look at the relationship between health outcomes and PFAS exposures. We're looking at a variety of health endpoints in this study, including endpoints associated with lipid metabolism, kidney function, thyroid disease, liver disease, glycemic parameters, and immune response. This is not a cancer study, but still a very important study as we to help us better understand how PFAS exposure through drinking water can impact the health of communities. Our pilot for our multi-site study is the PEAS study. And you will see on the right, the 7 sites. Our goal is to have about 1,000 adults and 300 children in each community, with 7,000 adults and 21 children total in the study. And finally, our P study, like I said, this is the pilot for the multi-site investigation. Our goal was to recruit 1,100 adults or older who worked or attended school at the Pease International Trade Port in Portsmouth, New Hampshire, anytime between January 2004 and May 2014. We're also looking to recruit 525 children, especially since there were daycare facilities on the trade port during this time period. And we're about half the way, 50% of the way through our recruitment process. And we still have about 6 more months of recruitment ahead of us. We're looking at both PFAS serum. We're looking at PFAS serum and urine concentrations for about 17 different PFAS compounds. We've already begun the process of conducting historical reconstruction and pharmacokinetic modeling of PFAS exposure. We're obtaining general medical history and blood pressure information. We're also conducting neurobehavioral testing on the children. So I previously mentioned some of the outcomes that we'll be looking at. In adults, we're focusing on endpoints related to cardiovascular disease, endometriosis, autoimmune disease, osteoarthritis. And in children, we're also looking at markers of sex hormones and neurodevelopment outcomes. So by the studies, I briefly went through this just a few minutes ago. We have, we're about halfway through our recruitment process. Right now, we're sitting at about 615 participants, and we have about 6 months remaining in the study. Some additional PFAS activities, like I said, we have a portfolio of work that we're engaged with. We have a collaboration with EPA looking at non-drinking water sources of exposure. So looking at things like household dust and how does that contribute to exposure. We also have work looking at the impact of PFAS exposure on viral susceptibility. For instance, what is the impact of COVID in communities who have high levels of PFAS in their drinking water? And is there a correlation between a greater impact of COVID or more serious outcomes in communities where we know the community members tend to have higher PFAS levels? Another large effort that we kicked off last year that we're about halfway through is we're partnering with the National Academy of Sciences to look at clinical guidance for PFAS and to really have them as a third party work with the physicians communities and other community members to come up with a set of guidelines for communities and community members who are exposed to PFAS. We're looking at previously collected data, doing ecological analysis to look at the association between PFAS exposure and cancer. And finally, we have some PBPK modeling work going on, especially as it relates to mixtures, because as we've seen in the previous presentations, nobody has just one PFAS in their body. There's typically different classes of PFAS. And so how do we model that in the human body? And then also looking at a water to serum calculator, so that if you have certain amount of a given PFAS in your water, how can we estimate what your serum level could be? And with that, other tools that we have online, our website is a great source for these. We have a PFAS only website. Our current clinical guidance is online. Our PEAT toolkit is online if you're interested in using that to assess exposures. Our tox profiles are online. We have various fact sheets. We also have a community stress resource center, and this is some work we're doing at ATSDR to look at the impact of environmental health hazards on a community's mental health and community resiliency. And PFAS is an area where we started to look at the impact of community stress related to PFAS exposures. And with that, that's all I have. Thank you so much for inviting me and allowing me to talk about ATSDR. And that's it.
Thank you, Christopher, for a really great overview of the activities you have going on. Just like in the first session, we're going to save questions for after the 4 presenters go and have a facilitated discussion. So we will move right to our next speaker, who's Stacy Anderson from the Health Effects Laboratory Division at NIOSH.
Hi, everyone. Can you hear me?
Yes, we can.
Great. Okay, so I'm going to briefly discuss some of my team's work related to the toxicity of PFAS following dermal exposure. So I'm going to switch gears a little bit and talk about some animal study work. So first, I want to highlight some of the research gaps related to dermal PFAS exposure. Well, most PFAS research has focused on oral exposure due to groundwater contamination. From an occupational perspective, the skin is one of the main routes of exposure. Additionally, occupational exposures are very different compared to those of the general public. Concentrations, addition of solvents, mixtures, and different formulations now influence exposures. While dermal exposure is an important occupational concern, there's very limited data related to the dermal PFAS toxicity, especially for some of the shorter chain compounds, and these with presumed less toxicity. So although most of you are very aware, I just want to quickly emphasize some occupations with potential for exposure. Obviously, firefighters and first responders, but also individuals in PFAS manufacturing, environmental remediation workers, someone has to clean up the mess, cosmetology, and fast food handlers are also occupations with high potential for dermal exposure. So I started researching PFAS, specifically PFOA, around 2005 when I started at NIOSH. My group has published numerous papers related to the immunotoxicity of PFOA in animal models. And I just want to briefly discuss these. So we did find that dermal exposure to PFOA penetrates mouse and human skin. So application of PFOA was directly on the ears of a mouse. And we found that penetration occurred through both skin of the mouse and human to more of a degree in the mouse. And also ionization state influenced the penetration through the skin. We also found that the dermal exposure on the ears increased serum PFOA levels. And there have been certain epidemiological studies that have associated PFAS exposure with allergic disease. And given that my group is a focus on immunotoxicity, we investigated this relationship a little bit more. And we found that dermal exposure to PFOA augments the allergic response to an experimental allergen, further supporting this association. A lot of the current literature has found that PFOA exposure is immune suppressive, but we also published a recent paper showing that it is also suppressive when administered to the dermal route of exposure. And also with this, we found dermal exposure influences alterations in skin barrier integrity and certain genes and mediators that were involved in this process, potentially looking at how this might be a mechanism for some of the related health effects. So more recently, we have identified some occupationally relevant PFAS, and these consist of the C4 to C7 series of the carboxylates and also the C4, C7 sulfonic acids. And this list of compounds have been identified as PFAS with potential for occupational exposure. So recently, within the last 6 months to a year, we've conducted some of the studies on the carboxylates, and we've done 28-day subchronic dermal exposure studies. So the animals received these PFAS on the dorsal surface of each ear. And we did these studies for hazard identification. So what this means is we selected the maximum concentration plus 2 serial dilutions that did not induce overt toxicity. And starting with the carboxylates, I just want to summarize some of the findings. And this is a lot, and I only had a short period of time here. So I just wanted to, I guess, describe these the best I can in this limited time. So what we did is we started with PFBA, which is C4. And we found a lot, following dermal exposure for 28 days, there were a lot of changes. And so we looked at a lot of different parameters, including organ weight, serum chemistries, histology, characterization of immune cell subsets in the spleen, lymph nodes, and skin. And then we did some more mechanistic approach looking at gene expression in both the liver and skin. We found some changes that were very similar to those that have been described for PFOA, which is listed at the bottom as C8. We didn't see some changes related to spleen weight and thymus weights. These are very indicative of PFOA being immune suppressive. We didn't see this with the C4 compound, but we did see lots of changes in the liver histologically and on a gene expression basis. The next compound we evaluated was C6, and based on what we saw for C4, we were actually expecting a greater degree of toxicity, but what we found is very similar changes, but to a lesser degree. So this sort of shocked us. We recently, last week, completed the study for C5, but we're still analyzing that data. From the study, we didn't see initial assessment. We didn't see changes, any dramatic changes anyway, in organ weights or serum chemistries, but we should have that data in the next couple of weeks. And then in July, we will complete studies for C7. I also want to mention that the data describing PFBA is in the process of publication submission, and hopefully in the next couple of weeks, it will be submitted for publication after clearance. And we've also collected some serum and urine from these animals, and the next presentation will discuss this a little bit more. So in conclusion, what we did found is dermal exposure does result in local and systemic toxicity. However, there might be different degrees and types of toxicity, suggesting different potential mechanisms. So the results that we found for these compounds were similar to what's previously been described for PFOA. However, the shorter chain length may not mean less toxicity. And this was, I think, obvious and evident with C4. It had more toxicity than some of the longer chain compounds. So there might not be a linear relationship between carbon length and toxicity, at least related to dermal exposure. So this does suggest the skin is a significant route of exposure. However, I want to mention there is also the potential for inhalation exposure and oral exposure due to the grooming of the animals. So we are considering this in future studies, and these results do suggest that additional investigations are necessary. That's all I have. Thank you.
Great. Thank you so much, Stacy. And our next speaker, also from the Health Effects Laboratory Division, is Dr. Cynthia Straley. I'll turn it over to you.
Okay, can you hear me now?
Yes.
Great. So now you're going to hear a totally different perspective of the PFAS from an analytical chemist. This project is a new project for me. I was not the driver of the instrument that I'm doing this method development for. And my mind is alive and lit up with learning this new LC-MS-MS technique. And I really, really love it. And when I found out that PFAS was the subject, I thought, wow, finally a chemical that I'll make a standard for, and the standard won't go bad in a week. But there was a different problem that showed itself with the PFAS. So, I'm sorry that happened. PFAS are everywhere and they last forever. Yes, I can make a standard for a PFAS compound and I don't have to worry about it going bad, but PFAS is everywhere in my lab. The chemistry lab has really bought into Teflon. All of our vials have liners with Teflon. There are Teflon filters. There are Teflon tubing. Teflon is everywhere. And consumer products, as we all know and been told, that those are another way that the materials can come into our lab from using lotions and products and having contact with things like Scotchgard and other kinds of water repellent surfaces on lab coats and just using them on your own personal self. So what had to be done was to adapt my LC to being a PFAS instrument. And that required, luckily, a kit has been made because this area of research is so expanding and popular in the scientific realm. There's a kit to adapt. You replace all your Teflon tubing with PEEK. You replace Teflon fittings with stainless steel. You use polypropylene vials in place of glass vials, since PFAS, not a contamination issue, but it does stick to glass, and you might not get a good injection of your standard or your sample. Also, LC and SMS grade solvents, really high grade, good stuff, is contaminated with some small amount of PFAS. And for each different vendor, it's a different profile of their background of PFAS in their solvent. Water was the biggest problem. Water from an HPLC manufacturer was definitely dirtier than water we could make ourselves with a millicube system, DI water with final filtration for chromatography. Also, using a delay column, which I'll explain later, helps you to move your background out of your analytical window. So these are the chemicals that we are looking at. And I forgot to mention that I am analyzing serum, urine, and potentially in the future organs from Stacey Anderson's mouse study for these different compounds. Nobody wanted to analyze compounds for PFAS that could be such high levels. A lot of labs do trace work, and they didn't even want a sample that high coming into their building. So we said we would do it since we're not doing any trace work, and started this process of developing a method. I found that of all these are the carboxy acids, 5 carboxy acids. PFOA was the most prevalent contaminant in all of our water and HPLC system. Also, the analogous sulfonic acids here displayed. PFOS was also the largest contaminant, and actually is the highest contaminant, higher than PFOA, in the water supply and solvents, and whatever is leaking or being extracted out of my pumps, my degasser, and all the other parts of the insides of the LC system. So this top peak on the chromatogram on the top is the first blank sample that I would inject in a day. I don't have it quantified, but it's like in sub, like 0.5 to 2 parts per billion. And that has built up on the head of the analytical column. And as you do multiple runs, and I should say that this chromatographic method has a gradient that washes the PFAS off the column. But if you keep doing consecutive runs, you do not build up any of this material on the head of the column, but just a little bit between runs. The chromatogram on the bottom shows you what you get from the addition of a delay column. We place the delay column in between the pump and the injector and the analytical column, and it gives us a few seconds of a delay, so that you can see here, I have a 6.5 nanogram per milliliter of PFOS, and next to it is the contaminant PFOS peak in my water. Electrospray MSMS analysis is a beautiful method and a beautiful technique. In all of the past of my mass spectrometry experience, I would just introduce a sample into the mass spec, and I would just read that mass of that chemical alone. But MSMS lets you selectively look at specific mass fragmentation of transitions, and that way you can transform a very complex and dirty looking chromatogram into a single peak that is only the analyte of interest. Here you see the fragmentation of a C8 fluoro compound. It loses one hydrogen ion and forms this charged species, which is called the parent species. Then it loses an SO3 group that forms the next daughter ion, and that we look at that transition to see our peaks. On the top here is a total ion chromatogram of a 10 all 10 PFAS standard, and to the right are the transitions of the selected parent ion for each compound to their daughter ion. And as you can see, you get a beautiful one peak chromatogram, and it's very specific, and it really helps with dealing with background, with dealing with things like matrix issues from urine extractions, blood extractions, serum, all those things that could be behind your peaks and not in just a pure standard mixture in methanol. Those often can obscure your peaks, co-elute, you have to change everything, and this takes a lot of that off the table. It's still possible that you can have something in your matrix cause problems, and you might have to change things a little bit, but it's really exciting how this works. Also, I'm currently, and here just to show you, I have successfully prepared the solutions and done my first series of calibration curves. This is a ULPC, it's ultra fast, it's a very short column. My runs are like 9 minutes, where I'm used to doing runs that were 45 minutes long in LC. So I can pretend like my background is doing immunochemistry and ELISA, where everything's done in duplicate and triplicate. This lets me do runs in duplicate, which is awesome as far as I'm concerned. So I can look at injection efficiency and figure out what my best analytical range is, and have more than just one run for my data. I'm currently awaiting for some internal standards that are mass labeled to use as internal standards for the solid phase extraction cleanup of the samples that I already have from Stacy Anderson from the C4 study, and be looking forward to getting the C5 samples. There's PFOA and PFOS. So all of my I'm not seeing any signal suppression. I'm not seeing any problem with this range of analytical compounds interacting with each other. Each compound had to go through an infusion phase where you infuse it into the instrument, you find its base peak, and then you look for a suitable daughter ion that has the highest value, and that's the most efficient. You get the highest signal. You just want the highest signal. So the ongoing work right now is to incorporate internal isotopically labeled standards. Many of them are in production by the company that I went to buy them from, but I already have labeled standards for the C4 and C5, and I have mouse sera, blank mouse sera that Stacy Anderson provided us. So right in the process of doing solid phase extraction and recovery of these two, and I'll have a background spiked sera run curve, and I will analyze those samples very soon. I'm going to analyze a small portion of the sample before this whole method is totally validated down to the dust, because I expect these to be really higher values than the lowest value that I can possibly detect. And I really need to see what my range is. Right now, I really have no idea. what the analytical range should be for these samples. So I'll be finding that out very soon. And I want to thank Dr. Jody Shoemaker of US EPA. She came and helped me out with figuring out where our contamination was. It was causing problems for the last chromatographer had the system before I took over. And she's a veteran PFAS analyzer, and she helped us get our system clean. And I've even got it cleaner, being more stringent with all the things I do and how I do them. I also want to thank the chemical and biological monitoring branch, all the people who have helped me do this project and help me do everything, and the biomonitoring team specifically. So thank you, and I'll answer questions afterwards.
Thank you, Cynthia. Our final presentation in the second session will be Dr. Suzanne Fenton from the National Institutes of Health. And I know we're running just a little bit behind, but we'll catch up some in the facilitated discussion time.
Okay, am I set to go?
You're ready.
Thank you again for the invitation today. This has been a great session. I want to talk to you a little bit about one of the emerging contaminants, GenX, or HFPODA. My slides aren't advancing, just 1 minute. Okay, here we go. So in North Carolina, you might know this already, we have quite a problem with these contaminants in the Cape Fear River Basin. Sorry. So it's been of interest in the state of North Carolina, New Jersey, and the Netherlands because we have fluorochemical manufacturers who are dumping this chemical into our water sheds. And the one, wow, I don't know what's happening to I cannot make my slides move. Sorry. Really sorry. I don't know. I tested it.
You tried the arrow keys?
I sure did. Yeah. Okay, here we go. So because many of the states and for health advisory setting levels, federal agencies and states are using the infant as the most susceptible population, we have conducted a whole bunch, several mouse studies I want to tell you about today, comparing the effects of GenX to PFOA. It's the chemical it's replacing in the marketplace. So right now, GenX is the new PFOA. So we expect that in the occupational studies that Miriam is going to be conducting, we hope she'll be able to get into some of the plants where GenX is being produced and let us know what the levels look like in people who get exposures more regularly than through the water supply in Wilmington. But one of the questions we asked and have published is what effects GenX may have on the maternal fetal unit. But today I want to talk more about what effects it has in the offspring that are allowed to be born and grow up. So we started these projects with 1 and 5 milligrams per kilogram of PFOA and 2 and 10 milligrams of GenX per kilogram GenX, mostly because the health advisory drinking water standards are double each other. So the EPA is 70 parts per trillion for PFOA, and North Carolina has a 140 parts per trillion level for GenX. So we just had them a factor of 2 apart. So we've looked at 2 different periods of development, and we have actually focused on outcomes that have been reported for lots of different PFAS. So we looked at blood clinical chemistry, we looked at cholesterol, lots of liver enzymes. We looked at the liver very specifically because so many PFAS have reported liver effects and lipid accumulation. We also looked at the offspring because so many effects of PFAS have been reported in children born to women in exposed communities. And we measured the internal burden of GenX and PFOA in all of these studies. Mark Streiner and Heather Stapleton are shown here as some of our collaborators on these projects. So just to summarize what we found in our first study of maternal infant outcomes, we found that there was gestational weight gain in the dams with either PFOA or GenX. We did find CITES accumulation in their abdomen, edematous tissues, higher liver weight. We also found increased placental weights and decreased fetal size. When we looked at the liver more specifically, we found that GenX was probably a more potent PFAS than PFOA on the liver because GenX doesn't accumulate in the liver. So we were able to measure the internal burden, showing that GenX was about 1/10 the level of PFOA given the same daily administration design only during pregnancy. And these animals were evaluated at the end of pregnancy. So in addition to that, 100% of the livers from both PFOA and GenX, both doses, showed some degree of like cytoplasmic alterations such as necrosis, hepatocellular hypertrophy, lipid accumulation. So we didn't have a NOEL in that study. We also saw, although the liver's effects and the clinical chemistry effects were very similar, there were placental specific profiles. So PFOA caused more congestion of the vessels within the labyrinth, which is where the trophoblasts are that control the growth of the infant. And the GenX caused atrophy of the cells within that same area. So they were different, and we did see differences in fetal growth outcomes. So we think that there may be different mechanisms involved in GenX and PFOA regulation of the placental outcomes. And we have transcriptomic studies ongoing right now to determine what those might be. But one thing I want to talk about today that's a little bit more important to this topic is the outcomes in the offspring. So we lowered the doses for this study. We went down to 0.2 milligrams per kilogram of GenX and 0.1 milligrams per kilogram of PFOA. And we compared those, we compared metabolic outcomes across the time span from birth until week 18 of life. We were looking specifically at weight gain, glucose tolerance testing, body mass composition, and clinical chemistries. One of the things that we noted in the animals in this study was, and this has been noted previously for PFOA, was that both PFOA, shown here in the blue for the females, and GenX, shown in green for the females, had a dose-dependent decrease in the developmental scores for the mammary gland. So the higher the GenX dose, the worse the development was in the mammary gland. When we looked at it at both puberty, peripubertal time point, here shown in the middle at PND 36, this dark spot in the middle is a lymph node, and it's really just used as a landmark because it's in the same place approximately in every gland. So you can see in the controls that the epithelium of the gland has grown way past the landmark, whereas it is not in the PFOA and GenX exposed groups. And these were effects that were present in both female and males, and they were dose dependent, shown down here in the bottom. We did a trend test, and they are dose dependent effects, specifically strong ones in the females. We also had our veterinary pathologists look at the mammary glands using just typical pathology with a longitudinal sections of the mammary gland at 18 weeks of age. And they were able to confirm that there was growth inhibition in the exposed animals that they did not see in the controls. In addition, there was probably some stromal hyperplasia and some abnormalities in terms of the way the fat cells were organized. So this is a persistent and permanent effect, it appears. We also looked at the weight gain of animals that were born to this study. Chemours had previously reported a decrease in body weight or birth weight in animals in pups in their study at 2 milligrams per kilogram or higher of GenX, and we repeated that finding shown here on the left. So GenX at 1 and 2 milligrams per kilogram decreased the weight of the pups in each litter when we did our culling of the litters. However, when the animals were 18 weeks old and over time, we saw an increase in weight in the animals, especially the males exposed to GenX. The same was true for the lower dose PFOA. We've seen this before in females. It wasn't quite statistically significant here. It didn't quite leave this line, but we did see a pretty strong effect in the two GenX dose group. Oop, sorry. Whoa, sorry. Another thing that we evaluated was the liver. We really were very specifically interested in this because these animals were only exposed to GenX during pregnancy. They weren't exposed during the rest of the time as adults. And yet, even though they were only exposed during pregnancy, it is pretty apparent that we have persistent and progressively worsening effects in the liver over time. We did see changes in cholesterol. We did see here shown in D this this foamy appearance, this glassy appearance that they it's from the lipid accumulation in small lipid vacuoles. We saw larger lipid vacuoles in periportal situation, which was worse in females than in males. So the females tended to have more of a liver adverse outcome, whereas the males tended to have more of a metabolic disease phenotype. And then also we had inflammation, periportal inflammation, which has been reported before for PFOA for GenX. All of this happened in animals that really didn't have any further exposure. And I want to just make that clear that although we measured PFOA, shown here in purples, on the left in serum, this is at culling, so this is at like day 5. And then we measured the GenX on the left in the kind of peachy colors. There's a large difference in the amount of these analytes in the serum, but we could see plenty of GenX in the urine of these animals on the right. We expected a bigger difference than this, I guess, because the half-lives are very different. In the mouse, it's about 3 weeks for PFOA, and it's about 1 day for GenX here. So we didn't really expect this. We think it's because it's an animal study. There's recirculation of the exposure due to grooming from the mom. But all of these exposures were gone for GenX by weaning. We couldn't detect anything at all. So in conclusion, liver homeostasis was significantly altered in, lipid homeostasis was significantly altered, not only in the liver, but also in the adipose tissue and in the mammary gland. So lipid homeostasis seems to be a real target for GenX. Mom and pup serum lipids were changed. There was increased body weight gain, body fat percentage. We also saw increased fluid retention and blood lipids in the male pups. Even though there was a slightly elevated glucose, there was a significantly increased insulin level in these animals by 18 weeks. In fact, we couldn't follow our normal protocol because they were so, we couldn't do insulin testing because they were so glucose sensitive toward the end of the study. So although we had planned it, we couldn't carry out that endpoint. Also consistent hepatocellular hypertrophy and lipid accumulation for GenX in animals that had no circulating GenX. The mammary gland effects for both GenX and PFOA were persistent, and we really saw changes in the fat pad and the epithelial mesenchymal transition in our toxicogenomics outcomes. So the next chemical on our list is 6,2-fluorotelomere sulfonate, which is found in both the contemporary AFFF that we're using in some of our other studies that hopefully we can talk to you about other time. And then it's also found in chrome plating, especially in the Great Lakes region. They have quite a problem with 6.2 FTS right now. So we are going to be conducting studies very similar to these on 6.2 FTSA in the very near future. Thank you. And here, thank you to the wonderful people who helped me do the study and to our collaborators. Thanks.
Thanks so much, Sue. We're going to go ahead and open it up for questions and comments. Christopher Ray had to drop off. He had a vaccine task force duties called him away. But for our other three speakers, if you'd like to either raise your hand or use the chat feature, we'll spend a few minutes having a discussion. Yes, Mark.
I have a question for Stacy. Excellent presentation. Thank you very much. It seems to be, and please correct me if I'm wrong, it seems to be a trade-off between maybe toxicity and bioaccumulation between the short and long chain isomers. And so in alternatives assessment, the big challenge for us is to, I guess, trade off toxicity and bioaccumulation, persistence, fate, and transport. So I wonder if you have any thoughts in regards to that.
I don't. We're just getting this data and we're just starting to consider this relationship. A good point, and it's something we'll consider, especially as we get the data for the serum and urine levels available to us. Something we've also considered was volatility of the compounds and how this may influence dermal exposure or, along with the chain length. So all of these things we are considering as we move forward.
Can I ask a question of Stacy?
Please do.
Sure, I'm here.
So Stacy, we have had to increase the kind of I don't know, the PPE for our people who are doing the dosing of our animals, because it appears that we can get aerosolization from the urine or whatever in the room from one dose group to the controls. Have you guys had any of those problems? Have you noticed that in your controls, or have you alerted your technical staff to these issues?
We haven't, and our compounds are delivered on the ear and acetone, so they're in a liquid vehicle. One thing that we have noted is, and I didn't put it in my talk because I only had 5 minutes, but we are seeing effects in the lung following exposure on the skin, and we don't know if this is a systemic effect or if it's indirectly due to inhalation exposure. So something we're going to explore more, which is really interesting, and we're really excited about that. So we saw it more for the C4 than we saw for the C6 with respect to the populations in the lung, which would suggest that, if there's a direct effect on the lung or if they're inhaling it, we're not really sure. We do house them in micro isolator cages. They are group towels, but we're not seeing effects, from the experimental groups versus the control, but we do have them together in groups of 5. So there would be the potential for oral exposure, from grooming and inhalation and all of those variables. So we are considering them as we plan future studies.
Yeah, we too have had to go to ventilated caging, and so we've eliminated a lot of it. But I do think this is something that everyone should think about in terms of, the occupation of the technicians that are doing your animal work, their safety. I think, really we need to think about this in terms of our animal studies in keeping those people safe while they're doing those studies for us.
Definitely.
Thank you. In the interest of time, we're going to move on to the third and final session. If you have additional questions or comments for our presenters, if you could use the chat feature and finish those there. We had a break scheduled, but since we're planning to end in about 20 minutes and I want to keep us on schedule and make sure that our final presenters have enough time, we're going to just move forward. Our third session is personal protective equipment and materials. And our first speaker is Rick Davis from the National Institute of Standards and Technology. Rick, I'll turn it over to you.
Okay, thanks. Can everybody hear me?
Yes.
Okay, good. All right, let me share this. This one here. Okay, can you see the first slide?
Yes, it looks great.
Okay, great. Thank you. So thank you for allowing me to present, and thanks for everyone doing a great job explaining PFAS up to this. It makes this presentation so much easier because I could really just focus on what we're doing. I also want to thank Miriam for realizing she left a timer on because I corrected that mistake before I got started on this presentation. So thank you, Miriam. So let's get started. So I'm Rick Davis. I'm from NIST. I'm a group leader, been there for about 20 years, and a project leader for a project that came our way as a result of Senate Bill 2525, which was co-sponsored by Senator Miss Shaheen from Massachusetts and Mr. Gardner from Colorado, which basically directed the director of NIST to conduct research associated with PFAS and firefighters by way of their gear. The funding arrived in March of 2020, and then the pandemic hit. So while it's a 3-year project, I'd say we're probably realistically about 6 or 7 months into the research. Let's see here. Whoops, move chat. So the project, in essence, is to conduct research aimed at reducing the risk of firefighters being exposed to PFAS from their gear. I just want to emphasize 2 things that have been highlighted. In terms of NIST, if you don't know who we are, we're a measurement science organization. So engineers, scientists, physicists, we focus on measurements. So it's really what's in the scope of reducing the risk is identifying the PFAS species, the concentration, the prevalence, where measurements are needed associated with the gear. We're not like some of the other presentations we've heard today. We're not health aspects, toxicity. We don't do that, right? So PFAS transport, human health effects, those kind of things, that's just not within the scope of what we do. So what I see is our research is sort of being that front matter, helping to identify what a firefighter could be exposed to, but what additional research needs to be done is how does that, those species and which of those species actually make it into the body of the firefighter? And then, as we've heard today with some of the research that's already going on, is what impact does it have once it gets in there? In terms of gear, we're keeping with the main ask that we got from Shaheen and Gardner, and as the jacket and the pants. We've expanded it to hoods and gloves because they're textiles, and frankly, they're made out of the same materials. So it's really kind of a twofer. If we find it in the pants, then in the jacket materials, we're likely going to find it in the hood and gloves too. Oh, come on now, let's go. Okay, so the main ask, all right. So the main ask with the from the bill is really to focus on the fabrics itself. We break this up in 2 parts. So new off the shelf, we do an extraction, we analyze, and I'll talk a little bit about what we do with that in the next series of steps. But what we're trying to get to is, in essence, what is on the gear that when the gear first comes out, is it a coating, is it a processing aid, those kind of things. But then we want to also then do some simulation stresses that would be similar to what a firefighter might see in the field: abrasion, thermal, laundering, and so forth. We'll do them as independent, and then we'll do them in sort of combination of each other. It's a combination of those 2 things that we could basically say, this is what a firefighter could be exposed to as a result of just their gear themselves. But I think there's some other pieces missing, and you'll hear that on the next slide. We've also expanded beyond that ask, and that is to include real gear. And I want to thank Miriam for helping us to find some gear that's from the field. That way we can understand, what is the sort of the reality aspect of what a firefighter's gear looks like. It's hard to say if we see something different in the simulated versus what's in the real gear, why those differences are there, because we're not going to really understand what that gear has been exposed to. And I really think a piece that's been missed by this main ask, and it's really a huge lift, and we're trying to lay the foundation for it, is that I think there's a real potential that PFAS could be found in the fire environment. I'm also calling it the fire scene. And so while that's beyond the ask, and it's a huge lift, I think we're going to lay the foundation with some of our work in this study is to understand, is there PFAS that's in the fire scene that could be making its way either onto the gear or into the firefighter. And this could be done through, if it's on their gear, perhaps it's dermal, maybe there's inhalation. If they're like me, they chew in their hands, so there is potential on oral transport. So understanding that. And NIST is uniquely set up in that we have large scale ability to do large scale burns. So we are going to build rooms, catch them on fire, and collect samples, air, soot samples, do wipes of the environment, put gear fabric in there and also test those. And we're going to start at a small scale with some bench scale to see, what products are going to have PFAS. If there's no sense in studying burning a kitchen, well, it might be fun. There's no sense in burning a kitchen if it's not a source of PFAS. So maybe living room and bedroom might be where we focus. We also want to get access to real fire scenes, and this could be by attaching sampling devices onto firefighters, and then such as SBE tubes, and we analyze them later. Also do wipes of stations, vehicles, gears, things like that, just to really get a sense of, is there PFAS around them beyond just the gear themselves. We're taking a target approach. That we got 72 analytes that we selected, and it wasn't just randomly selected. There's a very specific reason we selected those. So it's based off of literature, and it's also our experience working with PFAS and with textiles. We do have a plan on doing a non-target analysis. It's a heavier lift, and I don't know that we would get to it within the 3 years that this project is set up for, but we might. And if we do, it's going to be very preliminary type of work. So here's our 72, well, this is not the 72 species, but here's the classes that we've picked. We're using LC. We're also trying to get an understanding of the more potential higher volatile materials. So we're also doing GC. Our LC is a triple quad. We've got several other different instruments we're using, but we're likely to stick with the triple quad. And the GC, we're still trying to figure out exactly what instrument we're going to use. We have a sample prep done for both. And we're really comfortable with it based on spiking with standards. The GC was a little bit more difficult because they're more volatile. So blowing them down was a real issue, but I think we've got that nailed down. Here's some data that it hasn't been WRB approved, but it's been analyzed over and over again. It's been proven by 2 different labs, one that we have in South Carolina and our ones in Gaithersburg. So I'm really confident in the data. And basically what it says is that here's the moisture barrier, the outer shell, and the thermal liner, the 3 basic fabrics that make up the ensemble of a firefighter gear. There's PFAS. In the moisture barrier, it's primarily sulfamides. In the outer shell and the thermal liner, it's primarily the carboxylic acids. And again, this is off the shelf, just new. It's all LC, our triple quad data. And again, I tried to do as quick as possible, so hopefully I did that. So our research, again, I want to make sure everyone understands, we're focusing on identifying the PFAS species, concentration, and to what extent you could find it in the gear, where I think there's real gaps. And I've only been doing PFAS since the money came in March 2020, right? So you guys have a much better understanding of all of it. But what I see is the real gap is the understanding of the exposure and transport pathways of the PFAS, the health effects of it, and we're already seeing a lot of work here. So I'm really confident seeing that. Going beyond the preliminary work that we're doing in the fire scene and real gear, also thinking ahead, we already know that there are manufacturers that are trying to produce non-PFAS gear. I don't know that I really trust that it's non-PFAS. So I really want to analyze that. We want to see how that performance is not only new, which unfortunately a lot of the gear is, the service requirements are based on new, but what is the service life over time of that gear? And also cleaning. Yeah, there are a number of products that have marketed that they could potentially remove PFAS. I don't know whether I trust them since I don't know what PFAS is on the gear to begin with, but those are the kind of things that we could do. We'd also do it in probably in collaboration with MPPTL. We have a great relationship with them. Other things just to think about, NIST has fire grants, so we like to hand out money to help us do our research. And so if you are in an academic field and you have an interest in doing research with us, especially related to PFAS, I would encourage you to take a look at this website, and maybe there's an opportunity there. With that, bam, I'm done.
Great, thank you, Rick. And our final presenter for today is Crystal Forrester from NPPTL.
Okay, can you see it?
Yes, looks great.
Okay, thank you. So today I'm going to be talking about a project that just started in October. And it's very similar to the project that Rick just described that NIST is working on, but this one is working with laundering of firefighter gear and measuring the PFAS. So this current project is funded actually by NORA and began October 1st and is for 3 years. So our project is designed to test the hypothesis that durable water repellents, or DWRs, that are applied to the firefighter textiles could be removed during laundering and could possibly leach or deposit on the inner layers and cuffs that will provide direct dermal exposure to the wearer. So this project has 3 specific aims. We want to identify and quantify the PFAS compounds contained on the firefighter outer shell textiles. And that is actually changed to all the different layers. So this is actually a non-targeted study. We're going to assess the outer shells for PFAS degradation and removal during laundering. And then we want to evaluate possible exposures of PFAS to the wearer as the chemicals are removed. So when this project was originally proposed, our goal was to assess 1 to 3 outer shell textiles to determine which PFAS chemicals were present and their concentrations, and then measure their degradation through a series of laundry cycles. So this is now actually grown to about 26 fabrics that include 7 outer shell textiles with DWRs and 2 without that we're going to use for comparison purposes. We also have 5 moisture barriers, 7 thermal barriers, and 5 wildland textiles for a total of 26 different textiles that we're going to be working with. Some of these textiles are legacy textiles, meaning that they're not in production anymore, but are still being worn by firefighters because the gear typically has at least a 10-year life cycle. And then others are the newer textiles and may have the shorter chain PFAS compounds on them. So we have some specialized equipment that we're going to be using. The first on the left is our Milner extractor, and this is the same type of extractor that is used in fire stations and independent service providers that launder gear for fire departments. This particular model is programmable. It has an internal heater, so it will maintain the 105-degree temperature that's specified in NFPA 1851 on selection, care, and use. Maintenance of the firefighter gear. It also has limited number of G-forces. So the middle picture is our solid phase extraction unit. It's automated. This is going to really cut down our extraction time because it can extract 6 PFAS samples at a time, and it's specially manufactured for PFAS. And then on the right is our Thermo Vanquish UHPLC with a triple quad Thermo Quantiva mass spectrometer. So for our laboratory methods, as I said, we're going to launder everything in the Milner extractor that's programmable. We're going to extract using sonication, centrifugation, and solid phase extraction, and then analyze using mass spec, HPLC mass spec. And we are using the same extraction and analysis methods that NIST has developed. They were kind enough to share them with us. So the plot on the right is a sample of what we might expect a degradation curve to look like. However, so we're just getting started with this, so we're really not sure. They could be flat, they could have a steep slope, a gradual slope. So until we actually get this started, we won't know. But on the left is the laundry matrix that we're looking at. We're going to launder each piece of fabric for a certain number of cycles and then extract 10 swatches per laundering cycle, and we'll do that in triplicate. If we find interesting results, there's a possibility that we can use different temperatures. As I said, our machine is programmable, so we can go anywhere from 85 degrees up to 140 degrees. So as I said, our data is going to, our study is qualitative and quantitative. We'll use confirmation analysis through actual standards to determine positive IDs. And then we'll also use the 3 to 5 point calibration curves, blanks, spikes, internal standards for our measurements for the degradation curve. So we're hoping that we can determine what PFAS compounds are on here. We want to measure all of the PFAS compounds that the firefighters are exposed to. And we're hoping to identify these and then pass that information along to toxicologists who can prioritize their analysis to determine if toxicology studies need to be conducted, because if we're finding more of the short chain compounds on here, as Stacy said earlier, there's a possibility of adverse health effects with these. So we want to make sure that the compounds that are chosen or that are seen could be looked at a later time. And then this will also feed into information for the NFPA standards committees for inclusion in future standards. So I couldn't have done this without my team members, Matt Dooling and Jay Charlie. And I'd like to thank NORA for funding this project and our MPPTL leadership. And there's my contact information.
Great, thank you, Crystal. Let's just open it up briefly for some questions for Crystal and Rick.
Not everybody at once.
Hey, this is Susan. Can you guys talk for a minute about the fact that these are proprietary combinations and we're seeing shifting to the short chain and how that might impact what you're doing or what you're finding?
Proprietary in terms of the formulations or the gear or all the above?
Proprietary formulations for the gear.
Sure. Yeah, I think so. I think I'll answer for what NIST is doing. We're not looking at any heritage gear. Everything we're doing is new off the shelf. So, and we're not asking what is on the gear, right? It's quite frankly, I don't want to know. I wouldn't be able to disclose it. So our point is to take a very direct, suspect-driven approach, something that is very quick and easy that someone hopefully could pick up and do, so that as the formulations change moving forward, they can pick it up and do the analysis again as new things come out. Yeah, so I don't need to know what's in there, and they're not going to tell us, right?
Yeah, sorry, maybe I'll clarify. Because it's proprietary and you're talking about the methods development, could you talk a little bit about how that might impact moving forward when it comes to ultimately creating guidance or recommendations around the construction of the materials? I'm trying to link like where we are today and what we're doing now with what it could look like moving forward once we have the answers to these studies.
Yeah, I think that's very fair. So I think in terms of, and that's, well, what I have to assume, and this is not the area that we play in, right? We play in at developing and understanding what's there, but the regulations and standards that go along with that, that's not what we do, right? We don't get involved in that. So the only thing we could do is basically tell everyone what is in there. I think it'll be up to somebody to decide whether to force the hand of removing certain PFAS species from them. Now, what I think we've seen at a very limited amount of exposure so far is that manufacturers are willing to move forward with, some manufacturers are willing to move forward with a non-PFAS type gear. I don't know what that really means at this point. So at least there's an openness to do that. And I maybe as a point of reference, I'm also used to dealing with the halogen fire retardant issue where nobody wanted to change, right? The manufacturers did not want to remove it. So you needed regulations in place to drive them to do it. And in this case, we're seeing manufacturers willing to say, we'll take the initiative to move past them. Now, maybe that becomes a voluntary standard. I don't know, right? That's sort of out of the hand of where NIST would play, but we certainly would be willing to provide the science that goes behind any decision that's made in that field.
Thank you for the response. I think it's an interesting area, just looking at the conformity assessment framework and who the players are and the large engagement of voluntary consensus standards to look at how to move forward on this. And, you know, I also think, I think it was Miriam earlier made the comment where she said, you know, we have to really, or I think it was you, Miriam, maybe it was not, saying we really got to consider, you know, holistically what these compounds are helping to protect from as well, and do an assessment of what's the trade-off if you change the protections being provided. Are you exposing them to something worse or maybe potentially having a higher risk? And so I'm just also thinking through, there's a lot of work, I think, in the design of the materials themselves ahead of us to kind of address some of those questions. And so I think it's great work to get us started, and we've a lot of work still to do. Thank you.
Well, we are at time, so I would, oh, Miriam, I see your hand up.
I had a quick question, but if we're at time,
Sure. No, please go right ahead, and then we'll wrap up.
So I thank you for the talks, everybody. And I think I'm wondering with the studies of turnout gear, I know the focus of both of the studies at NIOSH and at NIST is on what's in the textiles themselves, what's in the gear as part of the manufacturing process. That's really primarily the focus, but there's also this component of what can adhere to the gear through the use and throughout the life of the product. Is there any discussion about how to evaluate how well all those potential exposures, whether it's from AFFF or from the fire ground or from just PFAS in water, how well those adhere to the gear, and then how well those compounds would be removed through various washing cycles?
Yeah, I think so I would think our ability to be able to do, so I think there's two parts. So if you recall, one of my slides was going beyond the main ask was to get gear from the field and analyze it. So this gives us a chance to understand what species are there and what isn't. And we can easily take the gear and wash afterwards, right? And I'd probably go to Crystal and ask for help on doing that. But I think there's also us being able to do, whether on small scale, sort of a bench, whether it be a cone calorimeter or a medium scale kind of thing, or doing a full scale test, we can put anything we want in there, burn, and then see what gets on the gear and what doesn't. So I think we have the ability to be able to understand, because we can test what products we're burning beforehand, what PFAS is present initially, what PFAS is present as we combust or heat up and then go to combustion of those products. And then what part of that is making it not only onto the fabrics, but again, because of our air sampling, we'll be able to see what makes it in as an aerosol. And I know there's got to be PFAS on the soot just because of the makeup of what soot looks like. So then analyzing what's stuck on the soot.
Thank you. And Rick, in the chat, Cynthia had asked about the link to the research proposal, if you have that handy and are able to share that. I'd just like to take a moment and thank all of our presenters today, as well as the attendees. I think this was a great session. I wanted to say morning, afternoon, depending on where you are, of some really exciting work that's going on in this area. So I just want to once again thank everybody for attending on behalf of Susan and I and the Public Safety Sector Council. We will be following up. The slides will be made available, as well as the recording to today's session, and then be on the lookout for additional meetings later in 2021 from the PSSC. We are looking at tentatively holding meetings in August and then in the October-November timeframe. So more details to come on those. Any final thoughts for today before we adjourn? Okay, well, thanks again, everybody, and have a great afternoon.